you hit that button, we'll do that. Hello, folks. Thanks for joining us today as we continue our digitalization webinar series sponsored by Novatek International with Digitalization of Stability Studies Management Statistical Analysis as our topic today. Our presenter is Susan Cleary, Director of Product Development at Novatek. And our panelists include Emily Dickinson, Director of Clinical Quality Assurance with Zentria, and Walter Routh, Founder of Sagana Consulting and Associate Editor of StabilityHub, and yours truly, John O'Neill, Founder of the Stability Discussion Group and Editor at StabilityHub. Just a few housekeeping notes for us. It adds a lot to the event if you can turn on your camera. We can deal with passing children and cats, and that's fine to be having your lunch while you're muted. Just the key there is to stay muted. Participants should stay muted unless invited by the moderator to unmute. It's okay if you need to clarify a question or make an additional comment. But in general, questions and comments can be submitted using the chat feature. We may also conduct polls through raised hands shown on our attendance roster. Just be ready to quickly lower them as soon as the count is taken so they don't throw off a later poll count. The meeting recording will be posted in the events section of StabilityHub.com, and the monthly newsletter article of the same title is available in the site blog section if you care to do some more detailed reading on this topic. With that, we'll turn the program over to Susan Cleary and encourage you to submit your questions at any point in the presentation as they pertain to the slide or topic being covered. It's all yours, Susan.
Okay, thank you so much, John. And welcome, everybody. Thank you for joining. Happy almost Friday. I'm going to hop right into it. So here we go. So what are we going to talk about today? So we're going to talk about what is digitalization. The elements of stability study statistics and how they fit in with digitalization, how the two, how stability study statistical analysis is enhanced by digitalization and what statistical tools can be available. We're going to talk about the why of it from ICH Q1E. And we're going to get into some details about what statistics you should be looking at for stability studies, including for individual results, your OOS, your OOT, your variability from a result perspective, and then patterns in the data, patterns across batches, across different storage conditions, and so on. Trending analysis, regression analysis, of course, data pooling. How do you look at your data? How do you pull it to look at it and ensure that your pooling is telling you that your data, your outcomes are more reliable because you're looking at the pooled data and you're verifying that it is poolable. Overall, we're looking at how we can alleviate statistical program risks and how to prepare for digitalization of your stability study data. And of course, we're going to go through some conclusions and benefits and outlooks for that. And throughout the entire webinar, we're going to ask for feedback and we're going to have open discussions and questions going on for all of the slides. So let's just introduce a bit of the topic. Okay, so when assembling a stability program, statistical plan is high on the list of requirements. You, of course, need to have the know-how on how you're going to analyze your data. You have to ask yourself, are you going to be compliant with the ICH Q1E? Do you have a sound plan and procedures for timely generation, creation of your data, entry of your data, and of course, finally, the analysis of the data that you're generating from your stability studies? What are statistical off-ramps into problem areas? And of course, also are our reports making the case for believable shelf lives with regulatory authorities? So are you producing the correct reports? Are you producing the correct statistical outcomes and graphs and supporting data to be able to submit to the regulatory agencies to show what your expiration dates are? So now let's look at some of the consequences, of course. So if you don't have the requisite stability tools, stability statistical analysis tools built in, then there are negative consequences to that. Of course, lack of trending is a big one, not being able to properly analyze your data, lack of approved statistical methods. So you can't just, pull pull out of the air a statistical tool, you have to know that it's relevant and applicable to stability data. Also, root cause analysis is important when you're looking at digging into your data. Reverse trace reports as well are complementary to your statistical analysis. If you don't have these, they may result in incomplete investigations, unable to find the root cause of out of specification values or out of trend values. Also, without the proper tools, erroneous shelf life prediction, you could be off in how you're predicting your shelf life, which of course is not good for patient safety. So we want to make sure that you have reliable shelf life prediction tools. Missing comparability opportunities. So there's so many different ways to look at data. And if you don't have the proper tools available to you, then maybe you're missing some options for comparability that would enhance the way you look at your data. Also, incorrect data pooling. If you're not looking at your data correctly and you're pooling the wrong batches from batch to batch, if you're not pooling them correctly, we're going to look later at, the hypothesis of, common intercepts, common slopes, common intercept, different slope, and so on. So really looking at your data, making sure that it is poolable to give you that more reliable shelf life. And of course, non-compliance, which is very important in this regulated environment. Did anyone have anything else to add about outcomes or consequences of not having the right tools available?
Only that from time to time you see some 483s that talk about a stats program where somebody did not trend their data at all or hadn't thought about it. And just outside of not having a stability program, period, I think that's the next best 483 that I've seen is that nobody does anything with the data that comes out other than throw it into a report finally when they register and don't really have any conclusions drawn from the data.
Yeah. Yeah, I remember, it's funny you bring that up, John. I remember working with a company one time and they didn't have any statistical analysis tools. You're right about that. That does happen. All they did was put it in a table where, they had all of the results and they just looked at it in a table with the time points, 0, 3, 6, 9, 12, and so on time points. And they just looked at it as a table without doing any analysis whatsoever. And of course, if we look at things nowadays, the digitalized solutions that are out there really provide for good statistical analysis capabilities. So I think everybody should have a digitalization solution in place. Now, one of the biggest questions I get asked is, what is digitalization? And, why is it different from digitization? And of course, digitization is just, entering data in an Excel spreadsheet, for example. It's digitized electronic files, electronic data. But really, digitalization goes so much further. These are more evolved and advanced systems. They transform the way you operate from a business standpoint. They harness powers of digital technologies, optimizing your process, in some cases, even controlling your processes. They increase your efficiency and enhance your decision making. And of course, in this case, it enhances your capabilities of analyzing your data. Now, I like comparing digitalization to Lego blocks. And you see my digitalization of my process on the right. We have our blocks. This is our data. And of course, we can sort it, we can arrange it, we can present it visually. But really, digitalization, true digitalization, explains it with a story. And your statistical analysis of your data tells the story of your expiration date. It tells the story of the products that you're testing and releasing to market. So really, digitalization provides you centralized access to your data, the ability to correlate across your data, high efficiency in analyzing the data and collecting it. Real-time monitoring, preventive measures, compliance considerations, all of these things are factored into a digitalization of your stability function. Now, we have done some webinars in the past, and I wanted to just bring in again about having the cornerstones of digitalization when we talk about stability study management. And really, shelf life and the statistical analysis is the part that you get out of the system. But really what goes into it is your data management and your equipment integration. And I just wanted to mention that these are the four cornerstones for digitalization of stability study management. And of course, risk mitigation is a natural outcome of having a system with data integrity and regulatory compliance and security built in. Data management, of course, is of the utmost importance because this is the data that you're going to use to do the statistical analysis. So having all the data available in that single system, okay, facilitates investigations, root cause analysis. Having an electronic notebook built in as well removes the need for paper worksheets, and the workbooks detail all the steps for executing an analytical method. So being able to efficiently capture the data recorded by humans. And of course, the second component, or one of the second cornerstone, is the equipment integration, having a really robust way to bring over the information from your equipment. And this is most important in digitalization efforts, that you automate the capture of this data. And of course, it involves equipment or other software systems. So our HPLCs, like Empower, we connect to the database, or, Agilent, we bring it over with file-based, lab solutions, or even just connecting to balances directly and bringing over the weights directly. So any different way that you can connect and capture data directly from your equipment will enhance your process immensely. We remove a lot of human error and improve the process and increase the efficiency and the reliability of the data. So for me, the equipment integration is one of the most important things you can do with your laboratory automation. And then, of course, shelf life analysis. Once you capture the data, you want to make sure that you're using that data to do proper analysis. And I always tell everyone, if you put a system in place and you have to export the data, then why did you put that system in place to begin with? So whenever you're looking about doing your shelf life analysis on your data, you want to make sure that the statistical analysis tools are actually built into the system. There's no export involved. The system is validated. You're working with validated data, and therefore your statistical analysis is also validated as well. So it's important to keep that data under the umbrella of the validation, the compliance that you put in the effort to create the system. Anyone have anything to add about digitalization, the cornerstones, or about the equipment, or anything?
Well, we have a question posed from Jeff Carr in the chat section, which again, anyone's welcome to post something there. But the question is the section in Q1E that indicates the amount of shelf life extrapolation that can be applied under different circumstances makes a number of references to the situations that are amenable or not amenable to statistical analysis. Can you advise on what sort of situations lead to data that are not amenable to statistical analysis?
I guess data that has failures in it. I mean, if you have data, thinking about it from regression analysis, if you have several batches and then you have a batch that you test and it's vastly different, it has a completely different interceptor, a completely different degradation slope, then I would say you'd want to exclude it because it's so different. And of course, that's something that would come out in a poolability test. Emily, do you want to maybe jump in on that a little bit?
Yeah, I will. I can contribute to that a little bit. As far as data pools that are amenable, the thing that I have found in my experience, I know for a fact, different conditions and different storage packaging configurations can also lead to data not being amenable. But then also to your point, Susan, with the degradation profiles, when we do stability, we do our frozen or our refrigerated conditions, depending on what the recommended labeled condition is of the product. We'll also do accelerated. Typically, your accelerated stability, where you're trying to stress the product, that's not going to be amenable to pooling because depending on the different parameters that you're testing during that study, it may be, I don't want to say necessarily a one-off, But depending on the type of product that you have and how much knowledge you have, it's not going to behave in a linear way that can be correlated back to how the product would actually degrade on the shelf. So I want to take a step back too, to your point, we were talking about, some of the circumstances and the tools that you can use for statistical analysis. One of the big ones that I want to point out is that product knowledge that you have from the very time that it's been developed all the way up through till you're doing registration. So really truly understanding how your product behaves in certain conditions will help to identify those situations where you can and cannot pull that data to get, whether it's you're trying to get a longer shelf life or, look at different possible storage conditions or, maybe not necessarily storage conditions, but, excursion, types of scenarios. So really I think the key cornerstones when you're going into doing a statistical analysis is truly understanding your product. And that's what you're going to get by doing those early stability studies. I hope I answered the question.
Yeah, thank you for that. So just to keep going a bit, of course, key components in good storytelling with your data, of course, is the summary of your statistics, okay, the central tendencies and the variabilities in the data to look at that, have the capabilities to look at that. Graphical representations for visual clarity, pattern detection, if you will, trends, your degradation profiles, time plots, scatter plots, and so on, which we're going to talk about a few slides ahead. Statistical modeling, like regression analysis for predicting future outcomes, outlier analysis. If you have outliers, maybe you want to drop them from your statistical analysis because they don't fit in. There was some anomaly with that data, confidence intervals to enhance the comprehensiveness of the reports, of course, and conducting investigations on your out of specs, your out of trends for, statistical elucidation. There we go. So let's talk a little bit about ICH Q1E and its significance. So it specifically addresses the statistical evaluation of stability data, establishing a framework that ensures the reliability of stability information. So for better decision-making for your shelf life and for storage conditions of pharmaceutical products. So, and while Q1E is the current gold standard for requirements, it doesn't get into specifics as to how to get there. And I think a lot of times the regulations do that. They tell you what you want to achieve, but they don't tell you how to achieve it. You have to come up with the means yourself. Really, we should be able to trace the starting point of our stats programs and procedures to the requirements of ICH. And this would be particularly valuable during inspections as the basis and justification for your practices and policies. And again, does anyone want to add anything regarding the Q1E and its significance to stability? Or we're good, we move on.
I'm good.
Okay. So let's get into the trending part. So trending on results. So when you're looking at individual results, we test our 0 time point, our 3 months, our 6 months, and so on. So we're looking for individual data points that break the rules and for broader patterns that indicate the product is changing in an undesirable way over time. And we're also looking for the point at which the product change is so great that it's no longer within expectations. So some of the criteria to look at is percentage variability. So, if you vary more than 5%, for example, from the initial time point, or if you vary more than X% from the previous time point. So just looking at individual results and their variability or their complete out of specification is one way to look at independent results. Change to the degradation rate, of course, is indicated by significant percentage change from the previous data point. So that's why we recommend to look at it. And, the rate of change of any parameter can indicate an adverse trend. Single results and variation in result-to-result trends can be visually highlighted at an entry point. So again, just bringing that back to our digitalization topic, when you enter a result in a solution, a software solution, you should be able to see right away, through color coding, is this X% different? Is it out of trend based on a trending tool like QSUM or Shewart? Is it out of trend, out of specification? So all of these visual notifications and also email alerts and notifications should be triggered just even for individual results at the entry point. Did anyone have anything they wanted to add on trending on individual results in a stability study?
We had a talking point come up that says it seems a new study with only 2 or 3 data points could artificially skew a trend chart. How far into a study should you get before including it in a trend report?
Okay.
I think this...
Yeah, I just wanted to say that usually when developing statistical analysis, I remember that the rule is 3. You have to have 3 points to calculate a line, and you should have 3 lines before you start looking at trending, so across 3 batches. So I think having, too little data is something that we need to look at, of course, and consider. And the rule that I've usually seen people apply is the rule of 3. You need at least 3. But again, if anybody else wants to comment on how they've done it or how they've seen it, that would be great.
I've seen the rule of 3 used as well in the laboratory, but I think that there's kind of a nuance to that rule of 3 too, that depending on how sensitive your method is or how fully developed it is, especially when you're in the early phases of doing your stability studies, sometimes if your method isn't finely tuned yet, you can get some artificial results. So one of the practices that I put in place in a past life was going out to 6 months, because then you can kind of see some of that wiggle waggle, start to, and that variability. Those wiggle waggle is my very technical term. But kind of start to level out, and you get to a point where if you do see those early shifts, they've kind of stabilized, for lack of a more appropriate term. But I mean, I 100% agree with the rule of 3, but I think that also needs to be taken as, it's depending on the situation you're in. It's, you're developing your product, it, kind of see where it goes, and especially, in some of those earlier studies.
Cool. All right. Thanks for that. Okay. We talked about individual results. Now let's just talk about looking at results together, and this is an example of a scatter plot. And of course, this is a great tool for comparing results, current ones with historical data. You can also, in this example, see, batch to batch comparison. You can see the x-intercept, and you can just see where they're landing. There's a slight downward tendency. So, new results that you get can be compared to existing patterns and reinforcing the degradation rate or showing a deviation from the expected pattern. See, our blue line is a little higher than most of the others. So, that might be something that you'd want to investigate a little bit deeper. Scatter plots support the identification of outliers and variation from batch to batch. Again, overlaying data from the multiple batches in one color and then adding the new batch in a different color. I've seen that done, or that's a tool that we have. So you have all of your previous batches, and then the new one is in a different color. So it really stands out if it's fitting in with the other batches or if it's popping out and doing something vastly different. And it visually gives you the capability of detecting that easily, and then you can go investigate further. So scatterplot is a valuable tool for identifying potential adverse trends and ensuring the ongoing stability of pharmaceutical products. Anybody have any comments or input with trends?
We had
Yes.
a discussion point that was raised about it's more a procedural kind of thing than statistical. Upon detection of a significant aberrant trend or trend result that threatens a spec at a future interval, what is a recommended chain of notification and elevation of the event? And thinking about that a little bit, I was thinking that the first person you notify is your supervisor, launching a lab investigation to validate the results, then elevating it to your manager, quality assurance, and then regulatory affairs for sure, because it'll impact the reports, and then finally on up to upper management. Any other panel members have, or anyone else have a different take on that or want to add to it?
As I say, in my experience, that's been the exact chain of escalation. A lot of times it's really critical to add an interim stability time point so you can see whether or not it might be maybe something that happened in the lab, or maybe that specific sample where you got some kind of not normal results. But that interim time point will then either confirm that you do have a batch that's going to be trending outward, or it can kind of lend back to that possible laboratory or some sort of anomaly with the sample. So the chain of escalation is definitely something that needs to happen. That way everyone's aware, when that interim stability time point comes up, that you're prepared if you do see a necessity to, if the batch is on the market, start that regulatory process of notifying the proper regulatory authorities. Or if it's a development batch, maybe there's something that happened in the process during that development batch production that could lend to that.
Thanks.
Another important principle would just be to make sure it doesn't appear, nothing appears as if you ignored a potential aberrant trend or something. So it's always, what's the agency going to think? And so you make sure you documented your notifications and your escalation, and they won't think that you ignored it.
I can tell you from experience, that is the absolute worst feeling, to be sitting across the table from an inspector and they point out a stability time point that you should have addressed and you did not. That's the kind of thing where your stomach goes up into your throat and you're like, I don't know. So yeah, preparation is key.
Yeah, exactly. And just again, going back to the digitalization and having everything automated, that blue line that we're looking at right now, the one that's higher than most of the other ones, in a digitalization solution, that would have triggered a notification to upper management and all the relevant people, so they would have been aware of it. It's not a decision just left to the analyst who's doing the testing and inputting the result. The notifications make sure it's escalated properly, and then you don't end up on that table in front of that auditor, trying to let them know why you didn't do that investigation that you needed to. So thanks for that. All right.
And one more point to that, I mean, that's one of the things when you're working in the laboratory as an analyst, you're seeing really one point in time. So it's up to stabilitarians and management to have that broader picture. So having those tools that make that picture more clear is definitely something that I can tell you, when I started in stability, I did not have.
Cool. Okay. So the next one, of course, is regression analysis, and I see this one come up often. So regression plays a crucial role in stability studies to determine shelf life and model the relationships between time and degradation, of key quality attributes. And of course, regression models predict how these attributes change over time. And once they're established, they're used to extrapolate and estimate the time at which product quality falls outside of acceptable limits. And of course, that's typically when you get to the 95% confidence band. And this estimated time becomes the basis, of course, for determining the shelf life. And regression also helps identify trends in the data, whether they're linear or nonlinear degradation patterns, and it allows stabilitarians to understand the mechanisms of degradation and optimize their storage conditions. And finally, regression analysis is additionally used to identify out-of-trend data points and that deviate significantly from the predicted regression line. And identifying these points is important to determine if they are caused by analytical errors or if they represent a real change in the stability of the product. So again, visually and statistically, regression trends can be used and be very helpful, in analyzing your data. And of course, that's, linear regression, but there's also multiple regression as well. These models, of course, look at the relationships between dependent and multiple independent variables. In the context of data pooling for stability studies and shelf life prediction, multiple regression helps analyze different factors affecting degradation rate of a product. And by pooling the data from different batches, which is what's going on in the example that you see here, by pooling them from different batches, a more comprehensive predictive model can be developed. And this approach enhances the accuracy of shelf life estimation by accounting for variability across multiple data sets rather than relying on a single data set that may not capture the full range of influencing factors. And what you're looking at here is several different batches that have been tested at different times, and they're all plotted together. And when you plot it together like this, you can look at your y-intercept, and start considering your hypothesis of common intercept. Excuse me, and you can look at their slopes and see if the slopes, which represents the rate of degradation, if their slopes are sloping at the same rate or degrading at the same rate. Does anyone want to add anything for regression analysis?
Well, I'm just curious. So this is the same data as the previous slide, and you had mentioned that the top, the newest data point would trigger some alert. Whereas if I look at the regression analysis, they look perfectly poolable. It's just, one. So my point is simply, this kind of highlights a question I have of false positives or false alerts. So if that previous slide, for example, triggered some sort of alert, however, you do further analysis and it turns out it isn't actually a significant trend, or it is poolable, it's consistent with all the other lots. How do you deal with that? Again, so you can't appear to have ignored it. You need to show that you looked at it properly. But one, how do you prevent too many false alerts and a waste of a bunch of time investigating? And two, what's the process in place to investigate false alerts? And does that make sense?
I guess the way I look at it is, you're going to have variability in the data, and having those notifications set at a point where, at a high enough percentage that you're going to detect, but not too high that you're going to detect too many false positives, is one thing you have to consider. So your percentage variability as one means of detecting them. So how sensitive your notification settings are, if you will. And the other thing I would think about is having multiple tools. Like we looked at one graph, and in one graph, you're right, it looks much higher. It might set off some alarm bells, but when we come here, we look at it, then yeah, you're right, it does fit in. It may be part of the same degradation rate. So having multiple tools available as well, I think, is important, and having the efficiency to look at these tools quickly. If you're dealing with an Excel sheet where you have to go and you see a notification, you see some alert, a visual cue saying you might have a problem here, and then it takes you days to investigate and look at your other tools, then I think that also is a problem and not being efficient, when you're looking at these potential false positives.
We have a question from the audience. Julie Liu has asked about what is considered a out of trend result from the regression line. I think from my experience, you can set a percentage that it's going to be considered out of trend, or as you gather more and more data, you can pull those trend limits in and be tighter as you go. So it would depend on your product and the results you've seen so far, or the procedure that you set up in advance when you don't have a whole lot of results. Any other comments on that? Maybe Emily or Walter?
I think what you just said, the procedure is key. I mean, you're going to have a procedure that says if it's a p-value of such and such, or your statistician can help you. But again, it comes back to that. The key, in my opinion, from a manager standpoint, would be to make sure that procedure doesn't result in too many false alerts, and a lot of running around for nothing. One thought I had as you were answering that question was maybe, some sort of a gradually increasing severity of an alert, so that the first alert is simply a trigger that invokes a more detailed statistical analysis, or something, so that it doesn't result in a lab investigation and a escalation just yet. So again, like John was saying, it all comes down to your procedures.
Yeah, I know in my experience, we've had internal both alert limits for this such a thing where you have an out of trend, but also internal action limits. So at what point do that action limit is typically your trigger point for an investigation before you actually go to the point where you have an out of specification. One other thing I would like to say about the regression line, which with regards to the percentage, that percentage is going to directly correlate to how much you know about your product, how stable your product is over time, and then how much developmental data you have. So one of the things that you need to keep in mind when you're looking at out of trends is what phase of development are you in? Are you in phase 1 or 2, maybe even 3, where you're getting ready to go into registration, or are you post-commercial? So keeping in mind, what is considered out of trend is kind of subjective. So it depends on a couple factors, where you are in your development process, if you've been commercialized, but also how stable is your product. So if you have a product that is going to degrade over time, your percentage for what is going to be considered out of trend may be a little bit wider, as opposed to a product that is rock solid. For example, I worked on acetaminophen, which is pretty rock solid over time. So when we would start to see shifts in the stability data outside of 0.5% or even 1%, we knew that something was kind of up. But knowing your product, knowing how shelf stable it is, can help determine what you're going to consider as out of trend.
Thank you. Anybody else?
Just a point that we're at 12:37, so a little bit past the halfway point for time.
Okay. So we looked at regression analysis, and you saw our multiple batches. So p-value analysis allows you to test if you can pool those batches. So p-value analysis is crucial for determining the significance of the independent variable in predicting shelf life. Okay, and a low p-value indicates that the variable has a statistically significant impact on the dependent variable, such as drug potency over time. In stability studies, p-value analysis helps identify the most critical factors influencing degradation and ensures only meaningful predictors are included in final models. It improves reliability of your shelf life predictions and allows regulatory agencies and manufacturers to make data-driven decisions about storage conditions, expiration dates, and of course, product quality over time. This is the test, the hypothesis test, where you say, are the slopes equal and the intercepts equal? Are only the slopes equal and the intercepts are different, or the intercepts are equal with different slopes? So there's 4 different tests, and we do the calculation here. And of course, it's calculated and compared against a critical p-value, in this case, 0.25. And of course, 0.05 that we saw in the text above. When you're thinking about a digitalized solution, you want to make sure that you can change your p-values and use different p-values for the comparison for the significance. So just having one value at your disposal is not enough. Make sure that you can change your p-value for the comparison and the outcome for the poolability test. And of course, if everything's pooled, this line should then become a singular line. It should give you the expiration date based on pooling all of the data you see here, not just based on one batch, one line. Any other additions for the critical p-value analysis? No? Okay. Now, ANOVA, which is analysis of variance. And this is a statistical test used to assess the difference between means of more than 2 groups. So it allows for simultaneous comparison of the arithmetic means across groups, and one can determine whether the differences observed are due to random chance or if they reflect genuine, meaningful differences. So again, this is comparing our multiple batches. It's the next step after linear regression or multiple regression. And of course, one-way ANOVA uses one independent variable, two-way ANOVA uses two independent variables. And by partitioning total variance into components, ANOVA unravels relationships between the variables and identifies the true sources of variation. ANOVA can handle multiple factors and their interactions, providing a robust way to understand intricate relationships. What is analysis of variance and how to use statistical analysis tools by Will Kenton. Of course, we're referencing his material. Emily, if you might want to add a little bit here, I know you're really good with the ANOVA understanding, so I think it would be very helpful.
As far as, so in my experience, sorry, my cat just jumped up on my lap. As far as looking at the different variables, one of the things that can be helpful when you're doing an ANOVA, I don't want to say run, because you're going to do multiple statistical analyses when you're looking at your data, but taking into account again, how is your product going to act at those different storage conditions, those stress conditions? So it's one thing to try to do that type of statistical model on, say, for example, a refrigerated product or a product that doesn't degrade, but once you start to stress it, so what exactly do those stress conditions look like? So are they impacted by the actual container that's going to be used post-marketing? So those are different factors where using this model can be really helpful to predict long-term how your product is going to react once it's in the hands of the patient.
Okay, thank you. Just to mention what we're looking at right now on the slide is you have each line on the upper grid here in the products. Each line is a different batch, okay, that we tested for a series of time points. And it gave, the trend description, so your linear equation, it gives the standard deviation, goodness of fit, and so on for each of the different lines. And then when you click, you see the top one is highlighted. You can see the intervals and the value for that batch. These are the values at 0, 3, 6, 12 time points, and so on. And then this was the predicted value based on pooling all of the other batches. And you can see here we look at the difference between the actual value and the predicted value. And this is maybe where you can start going and looking at, how my new batch, how far away from it is. If you get that notification, this is a place to go and investigate really how far it was away from that predicted value. So having a tool like this that you can go in and have all of these calculations done for you automatically, and then using it to investigate when you do have anomalies in your data, to find out really, is it a real problem or it was just a false false positive. So very important to have statistical tools dedicated for stability study statistical analysis built into the same system where you're housing your data. Next one, of course, is Arrhenius equation. And of course, Arrhenius equation is a tool for predicting the degradation rate of several categories of drug products under varying temperatures. And I think I always notice, for regression analysis, multiple regression, you're looking for that common slope. So you're using the same storage conditions, 25, or all your batches are under 25, or they're all under 30. So it's a common slope you're looking for because it's common storage conditions, so common rate of degradation. But with the Arrhenius equation, you can combine, your accelerated with your long-term and your intermediate. You can do it over variable temperatures. And of course, by measuring the degradation rate of the product at higher temperatures and applying Arrhenius equation, you can extrapolate the degradation rate at standard storage conditions. And this is crucial for accelerating stability testing process, saving time and resources while still providing reliable data. And of course, this was added that the Arrhenius equation can't be used when dealing with heterogeneous catalysis reactions showing Langmuir-Hinshelwood kinetics. Okay, so I don't know, Emily, do you want to jump in on this a little bit?
So a little bit of a story. The very first time I did a stability study using this type of model, it was, I don't want to date myself, but it was about 7 years ago, before it became one of the, I guess, the key tools in the toolbox for pharmaceutical development. I was, talking with my boss about how to, cut down on the development process for one of our co-development projects. And we decided to use this. We did not, one of the things, if you're going to try to do this type of stability study or this experiment, truly understand what factors could impact the degradation of your product. So is it going to be humidity? Is it going to be the temperature? Because the purpose of this is to actually push your product to degrade. You want to see that, but you don't want to push it so much that it degrades too fast and you don't get enough time points. So that's where you're going to partner with your development scientists that are working, in those dark labs with all the scary equipment that only come out, once or twice a day, covered in, who knows what. But truly understand, when you're setting up that experiment, what parameters you need to put in place to make sure that you're getting that degradation that you need to push it to failure, but not so much to where you're going to push it so far that at the very end you don't have anything to test. So it's kind of a fine balance, but it is very helpful if you can find that sweet spot, and you can get that data. This is incredibly helpful and really cuts down not only on the development aspect of your process, but also, if you've got a short timeline to get to a regulatory filing, it can help bolster some of those regulatory documents so that you can go to the next phase a little bit faster.
Cool, thank you. Anybody else? No? All right, and then finally, similarity. So what is similar and a statistical value between products? So is there a statistical similarity, comparability of different scales, formulations, presentations? Is there a statistical similarity of manufacturing sites? Is there a statistical justification for a bracketing matrixing strategy? So I guess, having the ability to filter and look at only data from a singular site, or only data for, specific presentations and formulations. And again, just looping it back to digitalization, having all your data housed in the one system and having these really robust filtering capabilities that you can easily bring in or remove data when you're looking at it in your statistical tools. I find is very important to be able to dig in and find these similarities and detect these patterns in the data to, really know more and more about your product and its degradation properties. Anyone else would like to add to similarities?
Just that it's a great tool when you're doing comparability studies, along with the poolability that we were looking at before. Lots of times we don't go heavy enough into comparability when we could actually save some resources by using the tools.
Yeah, I find now that we're, more and more relying on digitalization solutions, that it's not something you have to do that takes days and days, and it's in Excel, and you have to make all these tables in Excel and manually create the data sets and put them together and everything. Now that you have these really robust, advanced solutions available to you, I find people are more inclined to do these checks, of formulation, comparisons, presentations, similarities across manufacturing sites, because it's so much easier and so much time effective to do it now with the right solution in place. So just to, alleviating the risks and talk about benefits, I just mentioned more efficient. So, how does process control and digitalization benefit in relation to stability statistics? So it meets stability statistics needs and commitments. It reduces risks through digitalization. So you have more control over your data, more enhanced reporting and trending, data integrity, regulatory compliance, efficiency, faster time to market, and of course, as John mentioned in the beginning, no 483s, no FDA citations, of course. So really the benefits of doing things digitally now are really, really there. Of course, one of the biggest questions I get asked is, how do I prepare for digitalization projects? And of course, step one is identify the objectives clearly. So if your goal is to be able to statistically analyze your data in a digital solution, then that's your key objective. You want to put that down and define those requirements, as it says in step two, really define them, focus on your requirements, establish your new processes in line with the digitalization solution that'll come online. And then, of course, choose your technology wisely. Choosing a digital solution, you have to look at what you have now and how it can grow with you. So choose it wisely, because you can't always get what you want in the first go, if you will. Rome wasn't built in a day. So, you might have to take laboratory digitalization as iterations. You do a bit at a time, and over time, you work towards becoming fully paperless. And of course, always, as you put in new solutions, you're going to restructure to accommodate the change. And of course, execute your plans, allow space for agility. You have to keep up, and there's training involved. We always have that human factor, so allow for the agility. And then, of course, repeat the steps, to implement your next requirements. Would anybody else like to add anything for preparing for digitalization?
Could you hop back one slide, Susan? There was a couple of discussion points about the risk that had to do with, well, I'll read these here. When the pulling date from the same product has a lot of history and thus lots of variations in conditions, packaging, et cetera, how can you ensure that an automated statistical tool isn't generating a trend using unsuitable data? And in a hypothetical situation where an analyst is reviewing a potentially significant trend and determines that some of the data leading to that trend determination shouldn't have been included, is there a robust process for recalculating the trend without appearing like you're suspiciously massaging the data? So I guess a double-edged sword, some data could wander in that you didn't really want. And then if you're trying to exclude it, how do you do that without looking like you're at the foot of the fraud scale?
Okay, so the first thing you have to consider is whenever you're, we talked here about the similarities and manufacturing site scales, formulations, presentations, all of this information. So you want to make sure that any data that you're storing is atomic, meaning it's in one field by itself. And by doing that, it allows you to filter it and include it or exclude it. So, usually when you're looking at trends, the way I always see it done is you're sitting in front of your computer and you've got 10 batches and you start pulling in and out the batches and looking at them to determine really, are they poolable, are they not? You can pull out that different manufacturing insight and see what that does to your trending, or bring in all of the different presentations or pull them out. So just really being able to filter and dig into your data. And then if you do decide that something doesn't fit, that it's anomalous data and it shouldn't have been there, then really having the ability to prove, and justify why you didn't include it in your overall shelf life or expiration dating. So again, the efficiency of doing that process and the ability to justify your decisions really becomes enhanced by having all the data in a digitalized solution with the right tools to achieve all that. Okay, did that answer your question, John?
It does, thanks.
I had a quick, so earlier, Emily, you mentioned the need to essentially evaluate each product and its, like control limits, or, the way each product behaves. And then, Susan, you showed that ANOVA analysis on the screen that had various, that one, yeah. That showed those the predicted upper limit and lower limit, et cetera. My concern is you'd have to do that for each product and possibly each packaging. That's going to take a lot of manpower. So can you speak to that? So digitalization sounds like you're adding a whole other department.
No, digitalization means you're adding just one proper solution that does all this for you automatically and that's validated. So what you're seeing here is a real screenshot from an interface we have in a system. And this is literally, I don't know, I think there's 10 batches entered. And in seconds, it can calculate the predicted value, upper confidence, lower confidence, predicted and the calculated ones, the values, and it can display it all for you in seconds. So no, it's not like adding a whole new bunch of manpower. It's about making the people you have much more efficient about what they're doing so they can focus on other things and let the computer system do the math for them. I mean, that's what computers are really good at. They're really good at doing math and they're really fast and they're really validated. So yeah, this is a real system, and it really does get the job done very quickly.
Yep, those comments tie nicely into a question that just popped up. Is the test data entered directly into this program, or is it downloaded from current LIMS systems?
That's a good question. So what you're looking at now, it was entered directly into the system, but I have worked with other companies, and that's part of the reason why I really strongly advocate for having it in one system, because I have worked with other companies that don't have their statistical tools built in, and they end up having to export their data. And I've seen one company we worked with, we ended up building SSIS packages, which is SQL database programming packages for them to actually import the data into this system specifically just to use the statistical analysis. It's because they had a configurable system that I'll remain nameless, and that package has no statistical analysis whatsoever. So whether they exported it to Minitab, which is completely uncontrolled, or SAS or JMP or Excel or what have you, they opted to export it into this, which was a validated system where they could then use the reliable stats, with data integrity and with compliancy. So it can be done both ways. The data can be entered here. It can be brought in directly from equipment, or it can be brought in from other systems that simply don't have the tools that are required.
Have we run into any regulatory blowback from that particular situation?
No, like always, you have to make sure that the system is validated. So, you do your IQ, your OQ, we do our PQ, FDA's computer software assurance, which is kind of moving away from that traditional CFB with a lot of step-by-step testing, more CSA validation where we're actually testing the real processes. So as long as you're doing your due diligence and testing and proving that the interface works on a regular basis and that nobody can interact with the data during the transfer and so on, then you're covered from a regulatory standpoint.
Thank you. We have to say goodbye to Emily. She's heading out to another meeting. We thank her for her participation. And that brings us basically to our conclusion anyhow. I have a few announcements to close us out with. So if you have remaining questions, you're welcome to submit those to us. You can reach me at john@stabilityhub.com or walter@stabilityhub.com. And we can send those questions on to Susan and Emily as well if you need to. As we bring our program toward a conclusion today, we'd like to encourage those of you who are not yet familiar with StabilityHub to visit us at stabilityhub.com and explore the site for features that are the most useful for you and to sign up there for our monthly stability newsletter. Looking forward, we invite you to all to join us at 11:15 a.m. Eastern Time on Thursday, May 8th for PSDG office hours, 90-minute opportunity for you to bring stability questions of any nature, challenges and experiences as well before a widely experienced panel of stability subject matter experts. Whether you have questions or just want to pick up some pointers, you'll definitely come away with some valuable information. In June, parameter generation and control will bring us vital details for stability chamber validation, calibration, and requalification. And later in the season, Novatek will be back with another installation of stability management digitalization, some other aspect in which we can apply that. So watch for details in the monthly newsletter and in the events section at stabilityhub.com. I'd like to thank our sponsor, Novatek International, our presenter, Susan Cleary, panelists Emily Dickinson and Walter Routh, and you are a great audience for your attendance today, and we hope to see you again soon. With that, we'll wrap it up. Wish you all a great day.
Thank you.